Professor Subhash Chandra Lakhotia, sir, a distinguished Indian cytogenetics, renowned for his extensive contribution to genetics, cell biology, and developmental biology. Sir has been associated with Banaras Hindu University for several decades, serving as a professor in the Department of Zoology. Post-retirement, Sir continued his association with Banaras Hindu University as a Professor Emeritus and DAE Raja Ramana Fellow. In 2016, Sir was honored with the title of BHU Distinguished Professor, a position he holds for a lifetime.
All students and delegates, please keep quiet. This is one of the best lecture which we are going to be started. We have been going by listening, sir, and watching the presentations and everything. So please listen carefully. Please be patient and keep quiet. Thank you.
Beyond his teaching roles, Sir had also served as the Dean of the Faculty of Sciences and was the founder head of the Department of Molecular and Human Genetics. Sir's primary research focuses on cytogenetics, gene expression, and cell and the developmental biology, with significant work involving the model organism Drosophila melanogaster fruit fly. Sir's research has also explored the therapeutic effects of Ayurvedic formulations on neurodegenerative disorders using Drosophila models, providing insights into potential treatments for human disease. Throughout his illustrious career, Professor Lakhotia sir has received numerous accolades, including Shanti Swarup Bhatnagar Prize for Science and Technology in Biological Sciences, Fellowship of the Indian National Sciences Academy, FNA, Indian Academy of Sciences, FASC, and National Academy of Sciences, India, FNASC. Sundarlal Hora Medal from the Indian National Science Academy, J.C. Bose Award in Life Sciences by the University Grant Commission. In addition to Sir's research and teaching, Sir has been actively involved in shaping science education and policy in India. Additionally, Sir has served on various committees of National Scientific Organization, including the Council of Scientific and Industrial Research, CSIR, University Grant Commission, UGC, Department of Atomic Energy, DAE, Department of Science and Technology, DST, and Department of Biotechnology, DBT. Apart from it, Sir has been the editor of several international journals. Sir's dedication to science education is further exemplified by his involvement with the National Knowledge Commission and the International Council for Science, IECSU. Sir has mentored over more than 35 doctoral students and now has authored more than 200 research papers and numerous articles on higher education research policies and ethics. His enduring commitment to scientific research, education, and policy has significantly advanced the field of cytogenetics and molecular biology in India, leaving an indelible mark on the scientific community. So we are honored today to have Sir, who will share his insights on the topic, My Trust with Ayurvedic Biology and Lessons Learned. Sir, now we are eager to hear your thoughts and wisdom. Thank you.
Thank you so much. I hope I'm audible on the back. Is that okay? All right. So I first must thank Professor Vijayan and the Society of Biotechnologists India for conferring this honor, remarkable honor to me, and to make sure that I'm here. I appreciate the hospitality of this university, the faculty. I feel sad that the person who received me had to wait till midnight to get me, but that's something that we have to do with it. I definitely appreciate the enthusiasm of the young students who are around to carry it forward. And as Professor Balram Bhargav said, please remember those 5 points that he mentioned, because I can share you that this is what had carried me. These 5 principles have carried me where I am today. And that's very important, that we carry out our work sincerely, with integrity, with commitment, and so on, and then work on questions that interest you. That's more important. Original questions. Let's not be just followers. Let's also be leaders, which we can be. And I think if we can work within India, and I'm very happy when I'm talking about this, my lifetime work, almost whole of it is carried out in India with Indian money. And I'm very proud of it, that we have that facility, we can do it. And that's what I would want to share and enthuse young people that yes, we can do it. Now, keeping the philosophy of this meeting, I've chosen to talk about Ayurvedic biology. Let me first briefly say what I've been working with. Maybe these lights can be turned off, these stage lights. Then it will be much easier to see. So, I started my research in 1965 as an MSc student. And the first thing that we discovered was extranuclear DNA in a Myxosporidium parasite. And that led me to study genetics. And that's where my PhD research was on dosage compensation, and we showed cellular autonomy of dosage compensation. And that's where I got introduced to Drosophila model, the little fruit fly, which we now call as Cinderella of genetics, because such cute organism and a remarkable organ. And that's where my love for this fly has remained continuously till date. Then later on, I showed active transcription heterochromatin. Again, to young students, let me tell you, this was completely against the then dogma. Heterochromatin is inert, useless, doesn't transcribe. We showed it. The journal would not want to accept it, but then finally published because the evidence was very strong. And now we know that yes, that the whole concept was not always correct. We looked at replication in individual chromosome units and polytene chromosomes, beautiful giant chromosome. There we could identify each gene, what time it replicates in S phase and what order it follows. We did the DNA fiber rotography to look at the replicon size. Then during my PhD, I made a serendipitous observation on this 93D puff, which I followed up as an independent researcher. And in 82, when we thought that we will be able to identify what protein it makes, we discovered a non-coding. And that happened just 2 years after Craig and Ono and others have said that non-coding things are selfish and junk, which has become a textbook knowledge. And so for me, what should I do now? But I decided to continue. And I'm glad that I continued, because I never believed in non-coding being junk. I believe every part of genome has a function, and I'm glad that we did it. So we have continued, and this is what part, I'm not going to share it, but this has been my lifetime work. I'm continuing to do with this. I still have 3 PhD students working on this, and I hope I can decipher something more about it. And also, in 89 itself, I had predicted that non-coding RNA is going to be very important, which is now, you look up at any journal today, biomedical journal, cell biology, molecular biology, biotechnology, non-coding RNA, small or large, are everywhere. But that's where you have to have your conviction that yes, something that you're seeing is important. And so that's what we did. And then we identify the omega speckles based on this non-coding RNA, which regulate the very important cellular proteins, heterogeneous nuclear RNA binding proteins. They are dynamic in the cell, where they will be, how they will be distributed, and therefore it has very important regulatory role. Then I started looking at heat shock proteins, and then we discovered HSP16 Drosophila. When it was believed that there's no HSP16 Drosophila, we found it, and later on it was shown that yeah, there are 4 genes for this. And then we also showed that HRPA1 homolog has an important role in telomere integrity. And then we, about 10 years, 15 years back, I started looking at the role of these stress proteins in cancer development in Drosophila using the remarkable genetic that Drosophila allows. So we are doing that study. And then finally, the molecular biology studies on actions of standard Ayurvedic formulation, which is what I'm going to talk about. I mean, I chose to talk about this because this is a place where you're going to talk about Ayurveda, modern medicine, and biotechnology together. So I thought this will be something interesting for everyone to look at, this aspect. So this is my current work, non-coding RNA, stress protein, tumor development, Ayurvedic biology. And of course, beside this, as was stated, I write a lot about higher education, science policies, and more recently I've started writing on history of molecular biology. 4 articles have already come out in Resonance, and I think which is meant for students and teachers. It should be useful to bring us back how we are where we are today. What are in last 300, 400 years, what all things have happened. So this is something very interesting learning for me to read the history of things. Now, before I talk about Ayurvedic biology, let me make it very clear that I'm a basic researcher interested in genetics, gene expression, cell biology, using fly model without any formal training in Ayurveda or medicine. I have no training. I should also say I'm not a biotechnologist. In true sense, as one think of, I don't do any translation research or something. I use molecular biology techniques, but somehow students think molecular biology and biotechnology are same. No, they are not. And biotechnology is not just molecular biology. Biotechnology can go beyond Ependops and PCR and something. Any animal, any plant product, if you want to exploit, that's biotechnology. I would say even agriculture was the first biotechnologies that happened when we harvested the wild grasses into making our cereals and the domestic animals. So that's all biotechnology. Don't just think that only PCR or holding a test tube or running a gel makes biotechnology. No, it goes much beyond. But I don't claim to be biotech. I use those techniques for basic research. Now, my active interest in Ayurveda started with 2 names that were taken earlier by Professor Bhargav, M.S. Valyathan, a cardiothoracic surgeon, an innovator of remarkable capacity, and the other person, the atomic nuclear physicist, Chidambaram, who was at that time principal scientific advisor. They thought that we need to revive Ayurveda. We need to understand what Ayurveda is. And that's where they started the program called Ayurvedic Biology. And my interest in Ayurveda got into, because of them. I had interest earlier, but that was more of theoretical and general understanding. But this put me directly into it, and that's the authority on which I'm going to talk about whatever I'm going to talk about. And so my statements are based on a ring side view. I'm not in the center of Ayurveda. I'm just looking at it from periphery, and what I've experienced and what I've learned about it is what I'm going to share with you, which you can think, you may agree, may not agree, but at least I want a stimulus to be generated that people think about Ayurveda in a more critical manner. So when we talk about Ayurveda, we have classical books, Shusha Samhita, Charak Samhita. They're remarkable books, extreme detail, and at that time more than 2,000, which we do not know how they wrote, what they did, but they are remarkable in that sense. But the problem is, they were great scholars in ancient India. Where are we today? That's a blunt question that I wish to raise for young people, even senior people, to ask themselves, where are we today? Are we compatible to those great scholars? We take great glory, great pride in them, but we can't bask in their glory. We have to do it ourselves and see what had happened. And that's where important thing that I think look at is intellectuals in India are partially 2 groups: believers in Western philosophies and approaches, and faithful pro-Indians who like to follow ancient wisdom without any question. Now, this is important. I think that's where we are going wrong, that because they are ancient, we must follow it. We, as researchers, as knowledge people, we need to question things. We can't accept something just because it is stated by some authority at some time. And in the process, Ayurveda has suffered, negated by those trained in modern medicine, but faithfully followed by Ayurvedic practitioners and common men without any serious attempt to re-examine the old principles and practices in light of contemporary knowledge. And that's where I think meeting of this kind, where you want to bring it together, will be useful. And that's what I said, that we can't claim reflected glory of ancient sages as our glory. So what Ayurvedic biology planned to do, what was the objective when Valyathan and Chidambaram started this, to re-examine and understand the diverse Ayurvedic philosophies, practices, and formulations in light of our current understanding of nature, materials, and biological systems, to provide experimental and rational basis, and thereby sift facts and myths. And to me, this is very important, that we separate out what is fact, what is a myth. And that's what Ayurvedic biology hopes to do. And this only, in my view, it can rejuvenate Ayurveda where it should be. So my lab's objective in Ayurvedic biology, when I started about 15, 20 years ago, to have an unbiased inquiry into efficacy and mechanism of actions, molecular, cellular, and organ level of traditional formulations. And there we established Drosophila, my pet organ, because I have no experience in mouse or something human. I started using Drosophila, and we established Drosophila as a very good model, and now it is very widely used. Across the country, many people are using these organs to look at how things happen, because we have remarkable genetic facilities, genetic repertoire available in this fly, because this is only organism which I say can fly across continents without passport and visa. We share all the mutants across the world easily and without, there's some formalities involved now, but still they are easily shareable. And that's one beauty of this. So we published at that time 4 papers, research papers based on our work. I'll briefly explain this. I'm not going to get into details about this. The first we use our Amlaki Rasayan, which is, and Ras Sindhu, the mercury bus, and we compared the parameter like median lifespan, developmental time, salivary gland polythene level, fecundity, thermotolerance, starvation tolerance, oxidative stress tolerance, RNA processing proteins, and we found that there are some similarities, some difference between these 2 formulations. Now, for example, we looked at the levels of HNRPs in cells. If this is the control that normally growing Drosophila, but Amlaki Rasayan fed or Ras Sindhu fed, they have much more of this. We looked at individual genes. The polythene has the beauty. You can actually see in the microscope each gene. Which gene is active, which gene is inactive, and that's provided a great advantage. So we could see which genes, how much of this is present, this protein, and we could see that yes, when you give Amlaki Rasayan at the same gene, this becomes more. We confirmed this Western blotting as well. Then something that we unexpectedly found was, and this was, something you ask an innocent question. When we found HNRP levels are increased, and we knew that HNRP levels are known to suppress neurodegeneration. So we thought, okay, let's look at it, what happens. And then we made this remarkable discovery that these, both of them suppress polyQ and Alzheimer's and Drosophila. Now that's an advantage that Drosophila we can have human mutant Huntington gene expressed or human mutant gene that cause Alzheimer's. They can be expressed in Drosophila in a tissue where we want, at a time when we want, and that's where we could examine what happens, and that's the thing that we found. And the region that we found was enhanced levels of HNRP and histone acetyltransferase in these cells that express these 2. There's an increased proteasomal activity so that the toxic proteins are very rapidly cleared. Induced cell death is inhibited, so neurons don't die, and therefore you don't get neurodegeneration, and improved tolerance to cell stress, and thereby the cells can tolerate these toxic materials much more easily. For example, this is the way we do it. This is developing eye. I'm sorry. Okay, yeah. This is the developing eye, and this is where compound eye, where each eye has hundreds of eyes are developing, and we can express, because these are neural cells, the neuron that receive light, we can express the polyQ or Alzheimer's precisely in this region. And when we see in control, this polyQ is very high. Amlaki Rasayan, it becomes much less. Ras Sindhu is almost completely gone. Even Huntington, human Huntington with a 93 glutamic residues, also same thing happens. So this was an artificial model of Huntington. This is a real model of Huntington, and both things show same consequence. Similarly, the proteasomal activity, we use this repertoire, which will show higher GFP, proteasomal activity is less. Proteasomal activity becomes more, GFP will be reduced. And that's what we found, that Ras Sindhu reduces much more than Amlaki Rasayan, but compared to control, the proteasomal activity is very high. Then I collaborated with CCMP scientist A.P. Patel, who was working on mouse model of Alzheimer's and so on, and we found same thing happen. And S. Ganesh at IIT Kanpur, he has been working on Lakhotia disease, one of the epileptic disorder, and he also used these 2 and found that yes, even in mouse, these are working. And this was very pleasing, that what we see in Drosophila is also happening in mouse. And that's where I thought that this has implications for human health. And because what we thought was that Amlaki Rasayan and Ras Sindhu, because improve so many of physiological parameters that this is what Ayurveda says is healthy aging. That's what these rasayanas do, that they facilitate healthy aging. That was one. But then the important point was that they can become repurposed for effective therapy against neurological models, which are becoming very, very common today. And this is something that I think Ayurveda community need to take up more sincerely. And I wrote some articles and that this is something that should happen. And therefore, I think there's a need for systematic clinical studies on this. What we have shown in Drosophila and mouse, it has to be now tested in a formal way, because something that we need to know is Huntington's, and these are late onset disorders. What time we should start treatment? From young age, because today by techniques we can know who is likely to develop Huntington's, who is likely to develop Alzheimer's, because we know the gene markers. So once we identify them, and if we can have long-term treatment, can we suppress the debilitating system? That is something that needs to be done. So now I come to what I think Ayurvedic principles need to be re-examined, re-looked at, in what way they should be done. One, we understand that ancient scholars have a dynamic relation between life and matter based on their five senses. We have five senses and they looked at it. See, human mind, for that matter, any living system can learn from environment. We can learn more because of language, and therefore we can communicate and we can discuss and we can modify our inferences. And this, many things we learn by common sense. Just by observing something, we make an inference because brain keeps on working on that. And that's how I think they were very keen observers who looked at things critically, assimilated, and then they identify each of the five senses that an object has a specific property or guna, as we call it. And sensory perception identify the first, at that time, the five entities of bhutas, as we say, Vayu, Jal, Akash, Prithvi, and Tej, because these are things that we can immediately sense, and they are the most obvious things. If you don't go into details, broadly we can classify them, and that's what became the basis of Panchamahabhuta concept. And then, because as far as we know historically today, they didn't have so much of knowledge of physics and chemistry and biology of the system as we have today. Maybe we are wrong, but at least the evidence say that they did not have. So more of them will be from inferences, what they observed, what they knew, and what they assimilated and came to interpret them. And so that's where they talk about the five bhutas, the very combination of five bhutas generate the various forms, what we call dosha now. And that's what Charak and others who would have looked at human patients, individuals, look at their behavior, look at their body constitutions, identify, okay, this is Vata, Pitta, Kapha, three types. That's what they made it. They made subgroups. The problem is the bhutas, tanmatras, and doshas are influenced by ancient scholars on basis of their observations and the then available understanding material world. Now, they need serious reevaluation in knowledge, background, and light of what we now know about material science, about physical science, chemical science, about life properties. And that's where we need, now this point I have discussed in some of my articles. One was which were talked about that, and I think to me it's important that we need to demystify Ayurveda. And so we need Is it okay now? Then this is better. I think. Okay. All right. Sir, mic. All right. Well, why I don't like it, it goes far enough back and forth, and that becomes a problem. Anyway, I'll try my best. So that's what I think we need to do is, can we skip that point? It's not working now. Just press escape once. Now it's okay. Right. So I'm suggesting what we need to do is, this is something that I've been writing about for several years, but I repeat, we need in-depth, unbiased studies on ancient texts in synergy with scientific experimental studies. So what we need is participation of actual practitioners of Ayurveda with an open mind. Not that we want to prove something and therefore we do it. We just ask question, does it work? Does it do this thing? And if it does, how does it do? Or why it doesn't do? Both ways. Along with, we need linguists who can really understand the ancient texts in different languages. We have enormous number of old manuscripts lying around which have not been examined. Let's look at them. Let's see, even the Charak Samhita that everybody talks about, is it same as learnt in north, east, south, or middle of India? Are they same texts? Perhaps not, because most of these texts have been carried by word of mouth, and there's a likelihood of mutations happening. As genetics have to talk of mutations, deliberate, as we do it now today, genetic engineering, or random mutations. And so this is where we need people to examine these ancient texts in a much more depth manner. We need experimental scientists and modern medicine clinicians with expertise in fields like plant taxonomy, plant physiology, human physiology, cell and molecular biology, chemistry, material science, modern medicine, etc. Whatever you name it, we need them all to come together, as this meeting talks about integrated approach. And that we really need to do that. We need to understand mechanisms of biological actions of large variety of Ayurvedic formulations, rasayans, bhas, asavs. All of them are there. We still do not know what do they do, how do they do. And we need to do that. And that will require systematic and in-depth studies of mechanism of actions of these formulations at organism, cellular, and molecular levels. Understanding of the chemical, physical, and material science perspectives underlying elaborate procedures employed in preparation of various rasayan. Why do we have such elaborate processing? And this is something that I learned. Mercury, Rasindu, mercury sulfide is deadly poisonous. When we use the intermediate product, it was poisonous. But once it is finally processed, after all that grinding and it goes on, it becomes non-poisonous. But unfortunately, when I bought the Rasindu from several sources, some of them Okay, I'm sorry. This is something, problem of technology that we do it. But anyway, I hope it will be better now. Some echo is there. Can that be reduced? Okay, then we need to have precise identity of raw materials and preparatory procedures and stringent quality control. Because we still do not know what herb we are using. You get ashwagandha, so widely used, but I'm sure in different parts you can get ashwagandha from the market, which is not really ashwagandha. We need to be sure. How do we identify what it is? We need to understand seasonal and geographic variation. Like I remember reading somewhere in Charak book that the amla that you get in Madhya Pradesh, Central India, and in Himalayas will not have the same consequence.
Do we take care of that today? We don't. And we need to do that. Amla that we grow in rains or in winters, is it same? It will not be. And we need to understand what they are different and what difference it makes, which has unfortunately not been done. We well-defined process and quality control, because this is something where Ayurveda is really suffering. Formulation. There's no quality control. As I said, even I can say this, we tested few brands of Chyawanprash, which is commonly used. It's a household thing. Everybody almost takes it. Half of them turned out to be toxic to Drosophila. I don't know about humans. I'm sure they will be somewhere toxic. Why? Because there's no quality control. And that's what we need to do that. And I think biological parameters will provide much better quality control, because they're complex formulation. You can't test every chemical which is present and which is not present. Biological quality control will be defined that, okay, these are the consequences at cellular organ level. We can use that as a good parameter. And that's where this kind of studies become essential. We need to understand biological of Panchkarma, which is commonly used, but do we know what physiology, what immunological, what consequence they have, except hearsay? We need to do, there have been some studies, I don't say that there are nothing studies, but we need extensive, typical scientific studies in a rational manner. What are the physiological, immunological effects? How does it modify epigenetic regulation? Because epigenetics has now become very important regulation. What our daily life impacts what we pass on to next generation, that generation. Because there have been very, very many studies that my behavior, my experience, my exposure to certain things can affect my third, fourth generation, not genetically, epigenetically, and that's something that we need to understand. And I think we need to replace the dosha classification with contemporary criteria, because one is there have been several studies that Tridosha identification is not really rigorously done. Even my own example, I was attending an Ayurveda meeting, and one Ayurvedachari identified me as Vata type, and a few hours later, another one identified me as a Kapha type. I said, now, how do you treat me if I come to you? Partly because the criteria are not very well defined, they are overlapping, and that's where we need to do that. And something that we need to, for example, this paper came out from just 2 days back as a preprint, but they have been studying this many people have studied at genomic. So we need to look at genomic, proteomic, metabolomic, microbiomic, all aspects, because it's not just morphology that can tell us what is inside, how does it change. We need to do that study and then thereby identify what kind of treatment we need to provide. And then how do we train future Ayurvedic practitioners who are going to be future students who are here and other places? Besides learning fundamentals of Ayurveda, they should learn basics of contemporary biology, including cell biology, genetic development biology, biochemistry, physiology, and evolution. Because then only they will understand what life is, how does it change. They need to learn basics of material science so that significance and elaborate processing of rasayanas and bhasmas can be better understood. Why do we do this? Everybody today talks of nanomaterial, but do we understand why nanomaterial is behaving one way or the other? Are all nanomaterials working equally? We need to understand that. We need to learn contemporary diagnostic procedures because modern diagnostics is being used, but they should also understand why and how and when to use it, instead of just saying, okay, get ultrasound done and then come back to me. That's not the right thing. They must not be trained as dogmatic followers. To me, this is very important. They must be trained as questioning individuals who will question not only the patient, but also the treatment, why, how, and what can be done, and what's the basis of it. Historically, now, this is where we have problem, and which is where I think meetings of this kind can help bridge the gap, because historical modern medicine is based on identifying significant components in various herbs that have clinical actions, and their identification and chemical synthesis laid the foundation for development of contemporary modern medicine. This is what historically is true, that why Europeans came to India to first place. Portuguese came, French came in search of herbs, in search of spices, because they knew that they have clinical applications as well, not just test. But then they identified modern medicine happened, and we remain where we are. Unfortunately, that's something that I would say. So, and then what has now happened is because of reductionist approach, we look for active principle. I think this is not Ayurvedic philosophy. Ayurvedic philosophy doesn't work on active principle. Ayurvedic philosophy works on a holistic, integrated impact of whatever it is there. Everything is to be there. And every herbal preparation is not necessarily Ayurvedic. We often call it, okay, I'm looking at a herb and therefore I'm doing Ayurvedic research. Let's understand philosophy of Ayurveda first and then talk about it. And this is important to me in our understanding. And then there is a mutual skepticism between traditional and modern medicine. Ayurvedic practices are limited by lack of exposure to or training in comparative principles of biology. They are dogmatically faithful to the philosophies and principles of human body organization initiated thousands of years ago. I'm sure anybody who reads Charak's book, we admire that book greatly for its pharmacopoeia. But if you look at the physiology part of it, human body part of it, will not be very happy. But if you dogmatically follow it, because Charak said this, therefore we have to follow it, that's where we can go wrong. And so please be careful about this part. Practitioners of contemporary science and medicine, they are trained in reductionist and experimental approach. They are unable to correlate the principles and philosophies because they have learned reductionist approach and experimental approach, which Ayurveda do not do, and therefore there's a mutual skepticism, and that's what we need to break. And that can be broken if practice of Ayurveda need to come out of the shell which they have put themselves that, no, we know everything. What we have learned is complete. Let's not follow that. Even in modern science, we know that even something as like I mentioned, selfish DNA, every student of biology would have learned it in school, 1980. And by 2000, we knew that's not wrong. So if things can change so rapidly, how can you expect that something that was described 3,000 years ago can remain like a stone? It cannot. Actually, something that remains stone is dead. Actually, Mahatma Gandhi did say this, that Ayurveda practitioners, even in 1926, he said, it's dead science because they are not researching, they are not looking in a fresh way. And I think we need to change that. There's a strong need to correlate different languages and philosophies to find and understand the commonalities. The philosophical concept of Panchamahabhuta is neither experimentally viable nor compatible. I know many Ayurvedic people will be upset with me for making this statement, but I make this statement because I think there were great concepts of time, but we need to change them. We can't just go by their antiquity alone. And they should be learned by students only as historical facts, not as facts. That's my belief. I've discussed these in a number of articles. Now, this is where I would like to remind the teachers and students what Charak said about learning process. In one of his, the Charak Samhita Vimasan 8:14 number verse, roughly translated in English, it will read, the science of life shall never attain finality, which is a fact. Even today, with all the molecular biology, all the genomics and so on, we still do not know what life is. How does it work? We have so many things to keep on looking at it. Therefore, humility and relentless industry should characterize your endeavor and approach to knowledge. The entire world consists of teachers for the wise and enemies for the fools, mind it. If you think somebody talking and you don't like it, just because you don't like it, therefore you consider the other person as a fool or something, please don't do that. There can be some reason, as Charak said, therefore knowledge conducive to health, longevity, fame, and excellence coming from even an unknown source should be received, assimilated, and utilized with earnestness. If after all this you think it's wrong, it's wrong. But just by just saying, okay, this is against my philosophy, what I've learned, and therefore it's wrong, that's what Charak wouldn't have been happy about that attitude either. So learner and research must imbibe this philosophy. So what we need to do is integrate best parts of each healthcare system. Practitioners of modern and traditional healthcare system need to work together, since the larger objective in each case is to alleviate human suffering. And then only we can talk about Pachyema Shardha Shatam, Jivema Shardha Shatam, Shrunyam Shardha Shatam, Prabhavama Shardha Shatam, Adina Shama Shardha Shatam, Bhuyasa Shardha Satat. We want to live 100 years, but not a disabled person. Healthy aging, that's what the philosophy is. And that can come where all kinds of healthcare system can work together, can understand each other. And that's where what I said, scientists, linguists, experimentalists, the medical person, Ayurveda experts, everybody need to come together to re-understand and to reinterpret what we know about our system. So with that, I'm sorry for the wrong in norm. It's Biotechnological Society of India, or I miswrote it. But this is where I want to thank the society and Prof. Vijayan for the honor bestowed on me. It's really a great stimulus for me to continue. And I thank all my past and present students, associates for keeping me engaged with the enthusiastic approach to work. If I'm active today, thanks to these students who have been working, other associates group. And I thank, of course, this little organist for being an excellent model. And I thank you all for your attention. And I'll be happy to take questions if that's permitted by the organizers. Thank you. Okay.
Hello.
हेलो गुड आफ्टरनून सर सर हिंदू नववर्ष और नवरात्रि की आप सभी को हार्दिक शुभकामनाएं।
धन्यवाद
सर जैसे कि आपने अभी बताया कि हेट्रोक्रोमेटिक जीन जो होता है वो नॉन कोडिंग जीन होता है, तो मैंने कभी कहीं ऐसा पढ़ा है कि पहले पास्ट में ड्यूरिंग इवोल्यूशन पास्ट में वो जीन कभी फंक्शनल रहा होगा। क्या वो अभी कोई ऐसा जीन है जो हेट्रोक्रोमेटिक जीन है जो अभी तो फंक्शनल में है?
हाँ, हेट्रोक्रोमेटिन के बहुत से फंक्शंस हैं। इट्स वेरी क्रिटिकल। हमारे बॉडी में जो सेल में, चाहे ह्यूमन हो, चाहे मक्खी हो, चाहे बैक्टीरिया हो, नॉन कोडिंग रीजन जिसको हम हेट्रोक्रोमेटिन भी कहते हैं बिकॉज इट्स मोर कंडेंस्ड, हैज वेरी इंपॉर्टेंट फंक्शन, रेगुलेटरी फंक्शन। सी व्हाट वी नीड टू अंडरस्टैंड इन इवोल्यूशन इज for a function, we need a protein and we need a gene. But when, where, how much, how long that protein will be made, should be made, should not be made, this is regulation. And in that regulation, non-coding RNA, heterochromatin, chromatin conformation, they play a very important role. Okay, the old genes may have acquired new functions. Old genes, some of them have become pseudo genes, they still have function. They can have regulatory function. So nothing in the genome becomes completely useless because I believe natural selection, which selects for optimization of everything. And remember, in DNA, for every base addition, you need a triphosphate. And triphosphate is our energy currency. If in human genome we say there are 3.2 gigabases, and only 2% of the code for protein. Now imagine huge amount of DNA just carried, and we keep on spending our energy on that. I will never believe it, and now we know it's not true. They have functions. So that's what we have been also able to show, and now it's very well recognized.
Sir, somehow if we code the non-coding gene, can we use it as a beneficial or in the term of medicine?
Yes.
Healthcare.
Yes, because many of the non-coding genes are now turning out to be biomarkers. कौन सा कैंसर है, कौन सा non-coding RNA blood में, किसी प्रोटीन में ज़्यादा है, किसी सेल में, that can be a good diagnostic marker. और उसी को हम target भी कर सकते हैं. That if we reduce that, and this is very active research going on. But I think in context of Ayurveda, what we need to understand is, if let's say there are some drugs which have been suggested, formulation suggests for cancer, let's look at it. What do they do? Because one cannot be cured for all kinds of cancer, and that's where we need to examine them. And so non-coding RNA are very critical now for diagnostics as well as treatment targets.
Thank you, sir.
So it's still open for a few more questions, if any, from the students or participants. Yeah.
when we are using this Ayurveda, like global amount of study Ayurveda, how do we do this study? Because some Ayurveda formulation needs to be absorbed to digestion, but how to be applied locally.
Yeah.
So how do we solve?
Well, what we did was something that could be fed. Now, of course, you see, let me tell you, when I was presenting this project, other people questioned immediately, how will you identify Vata, Pitta, and Kapha types in Drosophila? We can't. That's obvious. But then we have different mutants, which have different behavioral, different body parts, but that's a different issue. But the feeding was an important thing. How do you apply them? Something that we can feed, how much dose to be given, we have to do trial and error. Because Drosophila, we can't, like, unlike mouse, we can't just put it in our mouth and it will go. It will feed at its own. So that's one thing that we do. Something that which is a liquid form you can inject. You can do cell culture studies. You can, if there's a vaporization product, you can make them inhale. So there are different ways. One will have to think out how to do it. Everything can be possible with Drosophila. That is very obvious. But so we have to find out how to do what best to apply, and one needs time. For example, in our study, we spent almost 3 years in ensuring how do we feed them, how much we feed them. And there's a project started in 2010. Our first paper came out in 2012 or 13. It takes time to understand that. Yes.
नमस्ते सर। सर मैं आपसे क्वेश्चन नहीं पूछना चाहूँगा, लेकिन सर आपसे ये बात करना चाहता था। मैं आयुर्वेद फील्ड से हूँ, बीएमएस का छात्र हूँ। तो सर जैसा कि आपने करंट क्राइटेरिया फॉर आइडेंटिफिकेशन बात पे कफ की बात की थी सर, तो सर आपने बताया था कि किसी ने किया तो वात आया, किसी अन्य ने किया तो कफ आया, उसको हमें देयर इज रिक्वायरमेंट, देयर इज नीड। बट सर मैं ये कहना चाहूँगा, जैसे ये जो करंट क्राइटेरिया है प्रकृति परीक्षण के अंतर्गत सर आता है। अभी तो एक बहुत बड़ा अभियान चला, प्रोग्राम चला। सर आपकी बात बिल्कुल सही है, पहले हमारे पास इसका एक कहीं ना कहीं प्लेटफॉर्म नहीं था, लेकिन आज की डेट में सर CCRAS के पोर्टल पे बहुत अच्छा प्लेटफॉर्म है, उसके माध्यम से ऑथेंटिकली किया जा सकता है कि आपकी प्रकृति क्या है। और जैसा कि उन्होंने, सर ने भी क्वेश्चन पूछा था कि मेडिसिन को कैसे करेंगे, तब भी आपने सर कहा था कि वात पित्त कफ का क्या यूज है। तो सर ये प्रकृति है, हर व्यक्ति की, हर इंडिविजुअल की एक प्रकृति होगी, उस प्रकृति के आधार पर सर उसको मेडिकेशन करना है, उसको आहार कैसे देना है। तो इस चीज के बारे में सर मैं कहना चाहता हूं।
मैं बिल्कुल मानूंगा, प्रकृति हर व्यक्ति की अलग होती है। दो भाई बहन, दो जुड़वा भाई या दो जुड़वा बहनें भी नहीं एक सी होती हैं। इवन इफ दे आर आइडेंटिकल, देयर विल बी सम वेरिएशन, जेनेटिक, एपिजेनेटिक, तो वो उसको आइडेंटिफाई करेंगे। लेकिन जो मेरे समझ में आया, जो अभी जो वात पित्त कफ को डायग्नोज करने का, वो एम्पेरिकल है। वी नीड मोर एक्सपेरिमेंट डेटा। भले वो कंप्यूटराइज कर दो कि ये आप कंप्यूटर से देख लें, ये क्वेश्चन पूछ रहे, ये आंसर यस है, नो है, but that's an average. It doesn't really tell you everything about that individual. We now have, although they are not very simple, but we need to understand that we need to do blind studies कि जो कि कुछ ग्रुप्स कर रहे हैं कि आयुर्वेद ने कहा कि ओके दिस इज वात टाइप या वात पित्त टाइप है, पित्त टाइप है, जो भी किया उसकी हम जीनोमिक, प्रोटियोमिक, मेटाबोलोमिक सारी स्टडी करें। उसमें 100% कोई नहीं होगा। What we'll learn is average, but that average will be better than what we are doing now. That's my point. I'm not saying that ये 3 प्रकृति गलत है, लेकिन उसी 3 प्रकृति में सीमित होकर रहने से हम नहीं ट्रीटमेंट कर पा रहे। और ये जो मैंने एक पेपर रेफर किया था, उन्होंने पूछा था, थ्योरेटिकल क्वेश्चन पूछे थे कई आयुर्वेदा लोगों से, अबाउट 500 सेम डिजीज कंडीशन, सेम डिस्क्रिप्शन, दे वर आइडेंटिफाइड डिफरेंटली, ट्रीटमेंट वाज सजेस्टेड डिफरेंटली। सो देन मरीज के लिए तो प्रॉब्लम है ना। सो जब तक हम अपनी ठीक से आइडेंटिफाई नहीं कर पाएंगे तो हम चाहते इफेक्ट वो नहीं होगा। एंड दैट्स व्हाट इज नेसेसरी दैट वी अंडरस्टैंड दिस।
थैंक यू सर।
Yes.
Thank you, Professor. Very excellent talk, and I like it so much. And it's not a question, but we experience something like this in Thailand too, because we have traditional medicine and modern medicine, and the doctors always refuse to use that. They cannot prescribe in the hospital in clinical settings. The main issue is about toxicity. They don't trust it, and also the efficacy as well, because there's no evidence to show to them that this would work in the patients. And most of traditional medicine, and the other problem in Thailand is about regulations. The Thai FDA or other things, the doctor have to prescribe only the drug that already registered and permitted by the FDA. So regulation to register is very, very hard, and it's not because they follow the guidelines of better medicines or other things. So the key is you need to identify the active ingredients, which is because the formulation of Ayurveda and others, also Chinese medicine, other things, is a complex of plants and other things. So it's very difficult to identify which actually the active one. Yes. So like you show in the slide, so what was your ideas, the approach that can use molecular biology to find the active target or the active compounds that could target in which signaling pathway or which defects pathway that could help, and then we can describe to the doctor, this is the mechanism, and you have to believe us.
Yeah, I appreciate the point that you are making. Like I can see the first part, traditional versus modern biology. I started as a traditional zoologist, learned comparative anatomy, and I didn't learn DNA when I was a student. I learned about DNA only later on. But today I'm so-called modern biologist. But then the important point, which I think what Charak said, learning has to be a continuous process, and which unfortunately today is not happening. Practitioners who are trained today don't want to either take it granted the old statements or just don't worry about, and this is where we need synthesis. That's one point. The other point that you mentioned was active ingredients. It is necessary to know active ingredient, but then what we are forgetting is, like you see, most of the modern medicines are derived from active ingredients. There are so many side effects. Why? Because firstly, we do not still know, we make all the docking experiment that this small molecule bind with this protein. Okay, it binds with it, but what else it can bind with? In the complex body that we have not looked at every molecule, that we still do not know this. But then the point is, when you're doing the docking, you're doing it in a pure protein, pure molecule, but body don't have pure protein. A cell is one of the most supersaturated medium. For a molecule to move, it has to encounter millions of other molecules. How does it change? And this is where the Ayurvedic philosophy of having a complex mixture of things, Anupan and so on, even traditional Chinese, they are doing individual component analysis. But then once you do that analysis, we need to come back and see when we combine them together, what effect does it have? And I think that's what is important. I know in China they have started massive movement. I was there in 2015 in one of the traditional medicine meeting, and I seen their lab, dozens of confocals in one room, dozens of HPLCs in another room, because they wanted to check every Chinese traditional Chinese medicine biochemically, molecularly, cell biologically. But then they must not stop there. They need to bring together, and that's what I think we need in Ayurveda or any traditional medicine, whether it be in African medicine, be it in Europe, some old medicines. We need to look at that, because traditional knowledge is remarkable knowledge. But only if we encash that in a proper way, instead of blindly following it, blind following has a danger. What I'm saying is we don't ignore ancient knowledge, but we need to re-understand that, remove what is not correct, take what is our current understanding, makes us learn it better. That's my point of view. Thank you for making this.
Thank you, sir. For your words have not only enlightened us, but have also inspired us to explore interdisciplinary perspective in science with an open mind. Once again, thank you, sir, for gracing this event with your presence and for sharing your invaluable insights. It was indeed an honor to have you with us, sir. Thank you. With this enlightening session, we now break for lunch.